Welcome back, everyone. Ready for another deep dive?
Always ready to explore.
Today, we're going cellular, zooming in on the incredible process of DNA replication.
Ah, yes, DNA replication, the heart of inheritance.
We all know it's vital, but I think the how is even more fascinating than the why. And that's what we're here for today.
Absolutely, unraveling the mechanisms behind this fundamental process.
We're going to be using Chapter 11 as our guide, the one on DNA replication and recombination.
A great chapter packed with details.
We're going to hit the highlights: semi-conservative replication, the roles of those incredible enzymes, and the added complexities that come with being a eukaryote.
It's like comparing a bicycle to a high-performance race car, right? Both get you from point A to point B, but the engineering under the hood, that's where the magic lies.
Perfect analogy. So let's jump right in. Semi-conservative replication, it might sound like a term from a political debate,
It does have a certain ring to it.
but it's actually the core concept behind how DNA copies itself. It's not just a simple photocopy, is it?
No, not at all. Every new DNA molecule ends up with one original strand from the parent molecule and one brand new strand.
Oh, that's interesting.
Like a dance where each partner brings something old and something new to create something unique.
I like that. A molecular dance. So this idea, was it Watson and Crick who proposed it?
It was a revolutionary idea that changed our understanding of heredity forever. Every cell division, it's passing on a piece of the original blueprint. Imagine that continuity across generations.
It's mind-blowing, but how did scientists actually figure this out? We can't exactly watch DNA replicating under a microscope.
Exactly. We had to get creative.
I bet.
They used radioactive isotopes to label DNA and track the old versus new strands. It was like giving each strand a tiny glowing tag.
Oh, so they could see where the original DNA ended up.
Precisely. And by analyzing the patterns in subsequent generations, they confirmed that replication was indeed semi-conservative. One of the most famous of these experiments is the Mendelssohn-Stahl experiment.
Very cool. Little molecular detective work. But this intricate dance needs choreographers, right? What's actually running the show at the molecular level?
We call it the replisome, the complex molecular machine responsible for orchestrating the entire process.
Replisome, that sounds impressive.
It is. And in bacteria, the star of this show is DNA polymerase III.
Okay, DNA polymerase, that rings a bell. But I remember learning about different types of DNA polymerase. Why are there so many?
Good catch. It can be confusing. DNA polymerase I was actually discovered first.
Oh, really? I wouldn't have guessed that.
But it plays a supporting role, more involved in repair and cleaning up any messes.
Got it. So polymerase is like the stage crew.
Exactly, setting the stage and keeping things tidy. DNA polymerase III, on the other hand, that's our lead dancer.
Right.
It's the one responsible for synthesizing those new DNA strands, and it does so with incredible speed and accuracy.
So speed and accuracy, very important for a lead dancer.
Absolutely.
But even the best dancers need a starting point. Where does DNA polymerase III actually begin? How does it know where to start adding nucleotides?
Excellent question. DNA polymerase III can't just grab onto a DNA strand and start adding nucleotides randomly. It needs a little helper.
Oh, what kind of helper?
It needs a primer, a short sequence of RNA to get things going.
Wait a minute, RNA. I thought we were talking about DNA replication. What's RNA doing in the mix?
That's one of the fascinating quirks of biology. DNA polymerase III actually needs that RNA primer to give it a starting point.
Okay, that's interesting.
It's created by an enzyme called primus. Once the primer is in place, polymerase III can start adding DNA nucleotides one by one in the 5 to 3 direction.
5 to 3, why that specific direction?
That's just the direction it works in, kind of like how we read from left to right. It's a chemical constraint.
Interesting. So DNA polymerase III needs its RNA primer and follows a specific direction, but surely it can't do all of this alone, right? What else is happening on this crowded molecular dance floor?
You're right. It's a team effort.
I figured as much.
DNA helicase is there, unwinding the double helix like a zipper to expose those single strands so polymerase can access them. Then we have single-stranded binding proteins, or SSBs for short. They prevent the separated strands from snapping back together.
Kind of like those little clips you use to hold hair back.
Exactly. And all the while, DNA gyrase, a type of topoisomerase, is working hard to relieve the tension that builds up ahead of the replication fork. It's like making sure the dance floor doesn't get too crowded as the helix unwinds.
Wow. It's like a perfectly choreographed ballet, but on a molecular scale. This is getting pretty intricate.
It is, but also remarkably efficient. Remember, this is happening in every cell of your body, constantly. Billions of nucleotides copied with incredible accuracy, and it all comes down to this elegant interplay of enzymes.
Okay, I'm trying to keep all this straight in my head. We've got our replosome with DNA polymerase the third as the star, but then there are all these other supporting players like helicase, SSBs, and gyrase all working together to make replication happen.
Exactly. It's a remarkable feat of molecular coordination.
It really is. But before we get too lost in the details, I want to circle back to something you mentioned earlier, the leading and lagging strands. You compared them to runners on a track earlier.
Right. Remember how DNA polymerase can only synthesize DNA in one direction, that 5 to 3 direction we talked about? Well, this creates a bit of a logistical challenge when you consider that the two strands of DNA run in opposite directions.
Oh, right. So how does that work? How does DNA polymerase handle that?
That's where the leading and lagging strands come in. Imagine two runners on a track. One runner, representing the leading strand, has a clear path and can run smoothly in a straight line. DNA polymerase can just follow the direction of the replication fork on this strand, synthesizing DNA continuously.
Okay, that makes sense. So the leading strand is like a nice, easy jog. What about the lagging strand, the other runner?
The lagging strand is where things get more complicated, because DNA polymerase can only move in one direction, and the lagging strand runs in the opposite direction to the replication fork. It has to work in short bursts.
Short burst.
Yeah, it moves away from the fork, synthesizes a short stretch of DNA, then jumps back and does it again, like a runner who has to keep stopping and changing direction.
So the leading strand is a marathon runner, while the lagging strand is more like a relay race with lots of handoffs.
A great way to put it. Those short stretches of DNA synthesized on the lagging strand, those are called Okazaki fragments.
Okazaki fragments, I've heard that term before.
They're named after the scientist who discovered them. And to create a continuous strand, these fragments have to be joined together. That's where DNA ligase steps in the final member of our molecular orchestra.
So ligase is like the glue that holds everything together.
Exactly. It's the final step in creating a complete continuous DNA strand on the lagging strand.
This is incredible. I'm really starting to appreciate the complexity of this whole process.
It is complex, but it's a testament to the power of evolution, creating such a sophisticated system for replicating the blueprint of life.
And we've only just scratched the surface.
We have, but we've laid a good foundation. Now, are you ready to tackle the next level of complexity? Eukaryotic DNA replication? Here we come.
Okay, bring on the eukaryotic complexity. My brain is primed and ready. What makes DNA replication so much more challenging in these guys?
Well, think about it. Eukaryotic cells have way more DNA than bacteria, like orders of magnitude more.
Oh yeah, I guess that makes sense. More DNA equals a bigger job.
Exactly. For example, human cells have about 6 billion base pairs of DNA.
Wow, 6 billion. That's huge.
It is. Compared to a few million in a bacterium like E. coli, it's a massive increase in scale.
So how do eukaryotic cells cope with this extra load? Do they just have superpowered DNA polymerases?
They do have some specialized polymerases, but that's only part of the story. One of the key strategies is having multiple replication origins.
Multiple origins, what does that mean?
Instead of starting at just one point on the DNA molecule, eukaryotic chromosomes have multiple sites where replication can begin simultaneously.
Ah, so it's like dividing the task and working on multiple sections at the same time.
Precisely. Like a construction project with multiple work crews. This parallel processing dramatically speeds up the whole replication process.
Very efficient. But there's more to it than just the amount of DNA, right? Didn't you say eukaryotic DNA is packaged differently?
You're right. Another major difference is that eukaryotic DNA isn't just floating around naked. It's packaged into chromatin.
Chromatin, that's the complex of DNA and proteins, right?
Exactly. And those proteins, primarily histones, help organize and condense the DNA, but they also create a bit of an obstacle course for the replication machinery.
So it's not as simple as just unzipping the double helix and copying the strands.
Nope. Imagine trying to copy a book that's been intricately bound and decorated.
Yeah, that would make things tricky.
The nucleosomes, which are the basic units of chromatin, those have to be partially disassembled ahead of the replication fork to allow the enzymes access to the DNA.
So it's like carefully unwrapping a present before you can get to the good stuff inside.
A good analogy. And then, as the new DNA strands are synthesized, new nucleosomes have to be assembled to package the new DNA.
Wow, so it's not just copying the DNA, it's replicating the entire packaging structure as well. That's incredible.
It is. And there's another challenge unique to eukaryotes that we haven't even touched on yet.
More challenges. Bring it on. I'm feeling brave.
All right, but this one's a real mind bender. It's called the end replication problem, and it has to do with the fact that eukaryotic chromosomes are linear.
Okay, linear versus circular chromosomes. How does that cause a problem for replication?
Remember how DNA polymerase needs a primer to get started and can only synthesize DNA in one direction? Well, at the very end of the lagging strand, when that final RNA primer is removed, there's no way for DNA polymerase to fill in the gap.
Oh, I see. So there's a little bit of DNA at the very end that can't be replicated.
Exactly. And that means that with every round of replication, the chromosome would get a tiny bit shorter.
That sounds like a recipe for disaster. Like losing pages from a book every time you photocopy it.
Wow.
Eventually, you'd lose crucial information.
You're getting it. And that's precisely the problem. But cells have evolved a brilliant solution to this problem. They're called telomeres.
Ah, telomeres. I've heard of those. They're like the protective caps on the ends of chromosomes, right?
Exactly. They're made up of repetitive DNA sequences. Think of them like a buffer zone. They don't contain any essential genes, so when they shorten a bit with each replication cycle, it's not a big deal.
So it's like having a bit of extra material at the end of a shoelace to prevent it from fraying.
Perfect analogy. Telomeres provide that extra bit of protection for the ends of chromosomes.
That's clever. But if they keep shortening, won't they eventually disappear completely? What happens then?
Well, in most of our cells, the ones we call somatic cells, that's exactly what happens. Telomeres gradually shorten over time, and this is thought to be one of the factors that contributes to aging.
Wow, that's fascinating. Our DNA literally carrying a marker of time. But you said most somatic cells, implying there are exceptions.
There are. Certain cells, like stem cells and germ cells, the ones that produce sperm and eggs, have a special enzyme called telomerase.
Telomerase, tell me more about this enzyme. What does it do?
Telomerase is pretty amazing. It can actually add those telomere repeats back onto the ends of chromosomes, compensating for the loss during replication.
So it's like a repair crew that can rebuild those protective caps.
Exactly. And this allows those cells to keep dividing without losing crucial genetic information. That's essential for maintaining the regenerative capacity of stem cells and ensuring that future generations inherit intact chromosomes.
So telomerase is kind of like the fountain of youth for these specific cells.
You could say that. It keeps their chromosomes young and healthy.
That's incredible.
Yeah.
But if telomerase is so good at preventing chromosome shortening, why don't all our cells have it? Wouldn't that make us, like, biologically immortal?
That's a great question, and it gets to the heart of the complex relationship between aging, cancer, and cellular immortality. You see, while telomerase is essential for certain cells, having it active in all our cells would be a recipe for disaster.
Disaster? How so?
Think about it. Uncontrolled cell division is a hallmark of cancer. If all our cells had active telomerase, it would be much easier for cells to become cancerous and grow uncontrollably.
Oh, I see. So having telomerase everywhere would be like giving every cell the potential to become immortal, which could easily lead to tumors.
Exactly. And in fact, many cancer cells do reactivate telomerase, essentially making themselves immortal.
Wow, that's a bit of a paradox. Telomerase is essential for life, but it can also be a driving force behind cancer.
It is. It highlights the complexity of biology, those delicate balances and trade-offs that evolution has shaped over millions of years.
So we need telomerase to maintain certain tissues and ensure the continuity of life across generations, but too much of it can be detrimental.
Precisely. It's all about finding the right balance.
Okay, I think I'm starting to grasp the incredible importance of these telomeres. But before we get too carried away with telomeres and aging, I want to circle back to something you mentioned earlier, the diversity of DNA polymerases in eukaryotes.
Ah, yes. We've been focusing a lot on DNA polymerase III, the main replicator in bacteria. But eukaryotes have a whole team of specialized polymerases.
A team, like different polymerases for different tasks.
Exactly. Think of it like a relay race, where different runners are best suited for different parts of the track.
Okay, I'm intrigued. Tell me about these eukaryotic polymerases. Who are the key players and what are their specialties?
Well, first up we have DNA polymerase alpha. It's responsible for starting things off, lays down a short RNA-DNA primer, kind of like setting the stage for the main event.
So polymerase alpha is like the opening act, getting things warmed up.
Good way to put it. Then we have DNA polymerase delta. It takes over on the lagging strand, specifically handling the synthesis of those Okazaki fragments.
Ah, so it's the specialist for that tricky back and forth synthesis.
Exactly. It's well suited for that task.
Oh.
And then on the leading strand, we have DNA polymerase epsilon. Its job is to synthesize long stretches of DNA quickly and efficiently.
So polymerase epsilon is like the long-distance runner, smoothly churning out those nucleotides.
Precisely. And of course, accuracy is still paramount. Just like DNA polymerase III in bacteria, these eukaryotic polymerases also have those built-in proofreading mechanisms to make sure the genetic code is copied faithfully.
They're constantly checking their work, making sure there are no typos.
Exactly. They are meticulous editors.
This is all so incredibly complex. I'm starting to feel like we've only scratched the surface of this topic. There's so much to learn.
You're right, we have. DNA replication is a vast and fascinating field to study, and there's always more to discover. But even with all this complexity, the core principles remain the same. It's about faithfully duplicating that genetic information orchestrated by this incredible symphony of enzymes working together in perfect harmony.
It's a beautiful thing, really. When you think about it, this intricate dance of molecules happening countless times in every cell of our bodies, ensuring the continuity of life.
It really is. It's one of those fundamental processes that we often take for granted, but without it, none of us would be here. No life as we know it would exist.
And the more we learn about DNA replication, the more we appreciate the incredible elegance and efficiency of this process. It's a testament to the power of evolution and the remarkable complexity of even the simplest forms of life.
Absolutely. And the beauty of it is that there's still so much more to learn. Every new discovery opens up new avenues of research and deepens our understanding of this fundamental process that underpins all of biology.
Well, I think we've given our listeners a solid foundation in the fascinating world of DNA replication. From the basic mechanisms to the unique challenges and solutions in eukaryotes, we've taken a whirlwind tour through this essential process.
We've explored the semi-conservative nature of replication, the roles of those incredible enzymes, the complexities of chromatin and linear chromosomes, and the intriguing world of telomeres. It's been quite a journey.
It has, and I hope our listeners have gained a newfound appreciation for the intricate dance of molecules that makes life possible.
Me too. It's a reminder that even the seemingly mundane processes happening inside our cells are full of wonder and complexity.
Well said. Thanks for joining me on this deep dive into the heart of DNA replication. Until next time, keep those brain cells replicating those amazing insights. It really is amazing to think about all this happening inside every cell all the time.
It's easy to take it for granted, but when you zoom in, it's just mind-blowing. The precision, the coordination, the sheer elegance of it all.
We've covered a lot of ground today, from bacteria to eukaryotes, leading strands to lagging strands, primers to polymerases. What would you say are the key takeaways for our listeners?
Well, first and foremost, that semi-conservative nature of replication. I mean, that's the heart of inheritance, right? Each new molecule carrying a piece of the old one.
Like a molecular thread connecting generations, passing down the blueprint of life.
Exactly. And then there's the incredible accuracy of it all. Those proofreading mechanisms, the layers of error correction, they're constantly working to protect the integrity of our genetic code.
It's remarkable how evolution has come up with such a robust system. And speaking of evolution, we can't forget about those adaptations that eukaryotes have developed.
Right. They had to up their game to handle those much larger genomes.
Multiple replication origins dealing with that chromatin structure that's not easy.
No, it's not. And then there's the whole telomere story, which is just fascinating in itself.
Oh yeah, telomeres, those protective caps like the aglets on shoelaces. Who knew they were so important for aging and even cancer?
It's a whole other layer of complexity, but it highlights how intricately connected these processes are.
It's a reminder that biology is rarely simple. There's always more to discover, more layers to peel back.
Absolutely. And the more we learn, the more we realize how much we still don't know.
Well said. Well, I think we've given our listeners a pretty solid foundation in the world of DNA replication. From the basics to the complexities, it's been quite a journey.
It has. And hopefully it sparks some curiosity to learn even more.
That's what we always aim for, to inspire that sense of wonder about the world around us, and especially the world within us.
Because even the smallest things, those tiny molecules and enzymes, they hold the keys to understanding life itself.
Beautifully put. Thanks for joining me on this incredible deep dive into DNA replication. Until next time, keep those brain cells replicating those amazing insights.